This is a GCSE biology presentation on drug trials. In this lesson, we're going to go through the process of drug trials, why we test different drugs. We're going to mention some specific drugs and what they're made from, and we're going to bring in terms like placebo and double blind. So to get started, there are 8 statements. Each one is either true or false. You've got to read through all 8 and work out which ones are true and which ones are false. Pause the presentation, and when you're ready you can restart to check how you've done. Pause the presentation now. So in terms of drug trials, we have the first one is true. Painkillers relieve the symptoms is true. Painkillers work by killing the pathogen is of course false. Paracetamol, aspirin and morphine are all examples of painkillers is true. Aspirin can also lower fever is true. Most painkillers work by blocking nerve impulses is true. Penicillin kills bacteria and it's produced from mold is true. And drugs are now made on a large scale by the pharmaceutical industry is of course true. So what are sources of drugs? Where do we get our drugs from? Well, most modern drugs are made in labs, but originally most of the drugs would have been derived from plants or microorganisms. Classic examples are Alexander Fleming's discovery of penicillin. If you ever get a fruit and look at the picture in the bottom right hand side and you see that green white mold that's growing on fruit, believe it or not, that's actually the bacteria responsible, the mold responsible for penicillin. You also need to be aware that we have heart drug digitalis originated from foxgloves and that aspirin originates from willow. They are mentioned specifically on the specification for AQA, and it could be a 1-mark question, so it is worth taking a moment to learn those little facts about foxglove, about the willow and about the penicillium. New drugs. As I said, most new drugs are synthesized in a pharmaceutical industry used by chemists. They normally start from some kind of plant extract and then put it through a number of chemical processes. When we get a new drug, it takes a very, very long time to test that drug. We need to check for 2 specific things. We need to check that 1, it is safe to use, and 2, that it's actually effective, it's actually doing what it needs to. Obviously, we need to do a lot of repeats and we need to spread it around. A lot of people need to evaluate it to make sure that there is a high level of accuracy in the data. Now, recently, obviously, we've had the vaccine for COVID released, and people are obviously concerned about how quickly this has come out. What you've got there is a case of where pharmaceutical companies may have been working on 20, 30 or 40 different projects all at the same time. They've basically diverted all of their resources into this one project, and because so much money and so much manpower has gone into it is why they've been able to produce this thing at such a high rate. They still have to go through all the same safety processes, they still have to make sure it's safe to use, and they still have to make sure that it's actually effective before they can use it. When we are testing new drugs, we are looking for 3 particular things when we're testing a drug. We're looking for its toxicity, which is reference to its side effects. We're looking for what is called its efficacy, which is how effective the drug actually is. And the third thing they're looking for is they're looking for the dose. They're looking to work out what is a high enough concentration of the drug that it will actually work, but not so high a concentration that we have really bad side effects, or because again it's a financial industry, that we're actually wasting money. We want to give just enough drug to do the job. In terms of the actual stages of drug testing, there are 3 stages you need to be aware of. Stage 1 is what we call preclinical trials, and that's essentially where we're working with non-living organisms. It can be cells, it can be tissues, it can be computer models, but we are not looking at living organisms at that point. Living cells, yes, but not organisms. Stage 1, its primary purpose is to identify if the drug actually works and if there's any severe side effects. If a drug makes it past stage 1, it goes to stage 2, where we have what are also called preclinical trials, but this time we start testing on living organisms in the form of animals. The primary purpose at this stage is we are further testing for toxicity. We're looking to see if when we start putting lots of tissues and organs together, do we get side effects that we would not have seen had we just worked with cells and computer models. And the third stage is where we have human clinical trials. And it's where we start off with very low doses of a drug and gradually build up till we get a drug dose that's actually effective. We start with healthy volunteers. The reason we start with healthy volunteers is if we started with people with the illness, they could have compromised immune systems and all sorts of issues that if something went wrong with a drug, if there's particular side effects, they are much less likely to survive than a healthy volunteer. Once we've given it to healthy volunteers and they're happy with the side effects being low enough that it's acceptable compared to the actual condition or disease, then we start testing on people with the illness. An important note is that it's actually illegal to test cosmetic and tobacco products on animals in the UK. Now, while that sounds like a very noble thing, it essentially came down to the fact that people stopped buying things that were being tested on animals to the point where it essentially became law that if you have products from the UK that they can't be tested on animals, and therefore it's a good marketing tool because other countries were able to make these cosmetics and stuff much cheaper, but when they imported them into the UK they couldn't publish that they were not tested on animals, so it was a good way of promoting the market. In an exam, you'll be asked about what's called a placebo, or they'll say in a study some people were given the placebo. A placebo is a drug that looks, smells, tastes exactly like the real drug, but it does not contain the active ingredient. So whatever it is we're testing to see if it works, it is missing. Everything else is the same. You're not able to tell the difference in any way. You need to be able to use that term placebo. We can also talk about what's called a double-blind trial. And a double-blind trial is when the doctor who's doing the study and the patient don't know if the patient is being given the actual drug or the placebo. The reason for that is because given the patient the placebo is able to identify if anything changes that it was actually a consequence of the drug rather than them just thinking, oh, I'm getting a drug, I must get better. And also a case of doctors can give away non-verbal cues. So they weren't intentionally telling the patient they were on the actual drug, but they were actually given some subtle cue that would indicate to the patient that they were or were not on the drug, and as a consequence it was impacting on results. So most drug trials now are double blind. Last thing to finish off. If we have human clinical trials, they are paid. They pay volunteers to take the drugs. One of the things the volunteers have to do is sign a waiver to say if they die, well, it's okay because they knew they might die if they took this drug anyway. Some of these studies last for up to about 2 weeks. They are sleepovers. You have to stay in this lovely big building, and they have PlayStations and all sorts of lovely stuff to keep you entertained. And every so often they'll come in and take swabs, take blood samples, or whatever it is that you've agreed to. And they will pay several £1,000s. However, some people are arguing that this is just taking advantage of individuals who are in need of money. So the question is, should we allow human drug trials and pay people for it? Is there a better way? Just come up with your thoughts. There's no right or wrong answer. It's just your opinion. Come up with some evaluations. It would be even better if you can go off and find some research and some articles and some scientific studies to back up your statements.